What is the plant reactome? It is a database of plant metabolic and regulatory pathways with projections to over 30 monocot and dicot species. And I'll just highlight some different points of access on our homepage so you can understand how to move around within the platform. There are icons taking you directly to browse our pathway selections graphically. There are data analysis options that I'll be going over in a little bit, as well as a generic search box up here at the top, which will allow you to immediately put in information about your gene or pathway of interest. To give you a sense of the breadth of the data that we're currently covering, I'm going to show you the release summary for our currently released set of data. We have a mix of curated and projected pathway data in our database. The curations are done in the Rhizosativa reference species right now. We also have curation efforts going on in Arabidopsis, but we've not released that at this point. So what we have is a set of reference pathways from rice, and then using primarily compare data and some other sources of data, we create orthology projections to these other species you see in this list here. It's a mix of monocots and dicots, and you can see that we have that organized to talk about, to show our relative projection strength into pathways in this column here, and then reactions and in gene products in the final column. If the color is trending towards blue, it means that we have a higher projection saturation. If it's trending towards red, we have a lower projection saturation. I can point out some interesting things about the nature of the projection. In the case of soybean and wheat, for example, you'll see that the number of gene products is quite high, and that is related to the ploidy within those species, confirming some of what we know about the biology. Down here in Casalith, you'll see that those numbers are quite low. At the moment, we attribute this to probably under annotation on a relatively recently assembled genome. We would expect those numbers to improve over time within the research field. So that gives you a sense of what we're hosting in Plant Reactome. And now I'm actually going to take you through the web application so you can see exactly what we have to offer. So going through the Browse Pathways icon will take you to our pathway browser space. And here I'm going to highlight a few specific areas. Across the top, we have a feature bar which contains a link back to the home page, a species selector, which gives us a drop down to move through all of our reference curated and projected data. An area to change your display settings. You can hide the panel on the left, the detail panel on the bottom, or both of them at the same time. An analysis tools button, which I will come back to in a little bit, a tour if you'd like some detailed information about how this page is laid out, and then a small legend on the right hand side to describe the elements on our pathways. If you want to see a detailed legend, you can also open this up, and it gives you much more information about what represents within the diagram small molecules, proteins, complexes, etc., and where you can find reaction nodes, for example. So that's the feature bar. On the left-hand side, we have what we call the event hierarchy. This is how we are organizing our curated and projected pathways. You see we start with the very generic plant pathways listing at the top, and then we break that down into cellular processes, metabolism, and regulation. And if I go into metabolism and regulation, you see we have our pathways thematically organized by biological function. Amino acid, metabolism, carbohydrate metabolism, for example, biosynthesis of various kinds, as well as some signaling. And I'll talk a little bit more about our signaling work in just a little bit. So again, on the left-hand side, this is the event hierarchy. And I see that we've got a chat question from Lena. Thank you very much. What I'm going to do is go ahead and go through the presentation and demonstration right now, and I will come back to those questions. Just keep them in that chat window, and we'll be happy to discuss at the end. Thank you. In the bottom pane here, we have what we call the details panel. And when we have a pathway loaded up in the pathway section, you can walk through various subsets of data related to the pathway. So let me do that now. I'm going to pull up a particular pathway, ABA biosynthesis and mediated signaling. And you see in this space here, this is the diagram panel. Come to that in a moment. But in the details panel, you have several tabs that give you an overview of information about this pathway. You can go to the homolog pathways or the orthologically projected pathways for Rhiza sativa via this list if you want. If you go to the molecules tab, there's a breakdown of the proteins by UniProt identifier and the compounds, small molecules typically, at the molecular level. And we also have an expression tab that connects our data to the Atlas project at EBI. And when Atlas data is available for a particular pathway and species, we see baseline expression experiments here. And I will come back to that in a little bit more detail. The analysis tab is for looking at results when we do analysis. The processes list just gives you a breakdown of where in the pathway hierarchy this particular pathway fits. And there's a download section which will allow you to download particular data in different formats for the pathway that you're looking at. In the main section here, we have the diagram panel, and this is where you're going to do most of your browsing when you come out to the site, I would presume. You can again make that space bigger by using this item up here to remove the event hierarchy in the detail panel, and there are zoom options that will allow you to zoom in and look at these diagrams in more detail. You'll notice one thing that we feature in the plant Reactome is the presence of cellular locations. So we have container boxes representing the cytosol, the plastid, the nucleoplasm down in these orange bounded box areas. And we can identify activity that happens on the cell wall barrier and lipid barrier space as well. Bringing the other items back onto the screen, that gives you a little bit of an orientation of how the pathway browser works and how you can move around within it. One other strength of the plant Reactome that we're looking to build, have started on, and we're going to continue in, is adding pathways for signaling and regulatory activity. We feel like that this is a space that could be expanded on. So we have been doing in-house curation in signaling for things like stragolactone, ABA biosynthesis and mediated signaling, ethylene signaling. We also have pathways for auxin and brassinosteroid signaling, moving beyond just the metabolic space and into the regulatory space. I'm going to actually jump back out to the search feature now to show you another way to get to information on the pathway browser. In the search box on the upper right-hand corner of the site, you could put in a term of interest. Let's just use one of the suggested terms for the moment, cytokinin. And if I search for that, you get search results that you can filter on the left-hand side in a variety of ways. For example, it will allow you to filter by specific species. I can look at this in rice, or I can look at this in other species. We'll stick to rice for the moment. I can filter by type. I can look just at protein pathway reaction or protein set lists. I'm going to look at pathways, for example. If I filter by pathway, now I'm looking at just search hits for cytokinin in pathways for rice. You can also further localize it to compartment if you want. So then I could select, for example, an entry in the pathways, and you get more detail about that rice pathway, cytokinin zooglukoside biosynthesis. At the bottom, you're able to immediately connect to the orthologous events projected for this pathway and other species. It gives you the Go biological process, and you can link off to EBI and look at that Go description. There's also, this is the important part for navigating to the pathway diagram, there is a location and pathway browser section, and it will allow you to expand a tree where you can go into the event hierarchy and go specifically to the pathway that your search has hit upon. So if I do this, it will kick over to that particular pathway as a result of my search results. Now, I want to show some of the filtering capabilities. If I go back and say I want to look at this within maize, I can change the filter to focus just on the maze projections for this particular pathway, and I can go into those results as well. Then when I click on that item, it actually goes over to, it switched in the species selector to maze, and then shows me the same pathway for maze with the accompanying changes in the gene products or proteins that are associated with this particular pathway. In this case, for example, it talks about these members. Here are the Gramene identifiers for Z and maze that are homologs of our projected rice entities. Furthermore, if I want, I can link out to maze GDB for the gene that I am interested in, and if the data is available, it will load that data and show you additional detailed information for it. We have similar link outs to Ensemble for a variety of our genomes at ensemble.gramene.org. Okay, now with that orientation and a sense of what can be done within the pathway browser, I'm going to jump over to the main focus of today's talk, which is the analysis tools. You can get to this two ways. If you go back to the home page, you can go in via Analyze Data, or if you're already on the pathway browser, there's an icon at the top of the screen right here, and you can use this to analyze your data. And it pulls up an analysis panel with two main sections. There's an analysis tools at the top, which is focused on providing overlay visualizations on a variety of data that you may upload and compare. And then there's a species comparison section below this. We're going to start by focusing on these overlay analysis tools. It gives you sample data that you can work with if you want to see how it works. On the right-hand side, there are some buttons that will allow you access to identifier lists, microarray data, metabolomic data. The idea is that you can bring your data to Plant Reactome and upload it via the file choice above, or paste samples of it into this window, and then project out to rice or to all of our species, not individually, but as a collection. After you've pasted it in, and then you get a visual overlay of what that data looks like. Let's walk through a couple of examples of that. If I pull up a uniprot accession list, this is a list of simple sample protein identifiers, and then what it's going to do is it's going to locate these on our curated and projected pathway set, and I've asked it to just project a rice, so it'll actually just look at our curated set. The result of that is you see at the bottom of the screen in the details panel a results list that shows you how many entities were found out of how many total entities, and it further breaks it down by hierarchy through the thematic sets and eventually gets to the individual pathways where you have hits. It also gives you statistical values for p-value and FDR. On the left-hand side in the event hierarchy, there's also a very helpful way to see where you have a lot of hits. You've got numbers after the name of the containers of the individual pathways. It says how many of these proteins that you have supplied here appear on any particular diagram. So if I jump down into, for example, ethylene biosynthesis and signaling on the molecular overlay, you will see exactly where in these yellow boxes here. See if I can make that a little bit bigger for you. Those uniprot identifiers may be found. And if you hover over the box and go to the blue information tab, it will give you an option to display participating molecules. And that will give you a detailed list that will show you exactly which items were hit on the basis, in this case, of the rice identifier associated with those uniprot identifiers. Do the same thing over here. Partially filled boxes mean that it was able to find hits for a set of the proteins that are associated with this particular reaction, but not all of them. In this case, for example, we had hits for 4 out of 5 on the basis of the sample uniprot data that I uploaded. Sushma and Pankaj, I appreciate the running commentary in the chat window. That's very informative. Thanks. You can also use gene identifiers if we're going to do a different kind of analysis. We're going to clear this window here, and instead I'm going to use a file list. I'm going to get a set of MSU IDs to show the file upload option. If I can get my window out of the way. And I hit the Analyze button with those, and I get a molecular overlay that shows me, again, I could pick any number of places where I get a hit. Now this is going to have a lot of hits in this example because it's we just pulled out a sample set of gene products that we knew to be present in our curated pathway data, and you see all of the various places where that hits. Another example would be working with metabolic data. So if I were to go back here, and what I really need to do is get this chat window out of my way, bear with me. You can load up metabolic data even over multiple samples, for example, and you can analyze that data. In this particular case, it's going to give relative it's going to give you a color bar on the right-hand side that shows relative or presence or absence of those metabolites. And if, for example, you provided multiple samples or say a time course, this orange bar in the middle of the diagram will allow you to walk through that time course series where you can observe changes in value on the particular expression, the particular metabolite presence. You could also do this, for example, on all species. If I unchecked the Oryza sativa box, I wanted to look at metabolites across all species. I analyze it. It takes a little bit longer because it's going to be looking at all of our data now. And once that loads, you can see down here in the species name area, I'm able to see many different boxes, many different species, and I can go look at the examples of metabolites in other species as well. There we go. There's the color overlay, and I'm going to actually make that a little bit larger so you can see what I'm talking about. And there's some very subtle color changes in this particular sample data set that you can see as I cycle through the the numbered samples. Okay, now I'm going to go to a couple of other examples that we've pulled from literature, either publications that we have worked on or that we are just available out there and publicly available for rice data. The first example I'm going to work with is some diurnal data, so just to reference the paper without getting into too much of the, that's a drought stress, just a moment, yes. Without getting into too much of the biology, just to point out that some of our collaborators and people in our lab have worked on global profiling of rice and poplar transcriptomes, and they're looking at diurnal function and circadian control pathways. And so we took some of the data from that paper, and you can upload that onto Plant Reactome and see exactly where that appears. I'm going to switch back to rice, since that's the topic of discussion. Go to the analysis tools, clear the space, and what I'm actually going to work with is a subset of this data for the purposes of this online demonstration, because the analysis tools, when you work on a large data set, may take a while to return. They've got a very nice architecture in the Reactome software system that will allow a set of your data to be uploaded to the server, processed independently of the website, and then returned when it's ready. It lets you wait in the window, but it takes a little bit too long for the demo if I'm using a very large set of data. So I'm going to use a smaller set of data for this example and actually paste it in the window. This is a subset of the diurnal data, so it's not at all representative of the coverage that you might see as a result of this actual experiment. But it gives you an idea when I project a rice that there are indeed hits for a particular experiment, and you can go up to an upper level pathway, and this is something I wanted to show as well, that it will actually show you presence or absence of the number of entities in your expression analysis at the thematic level. So you could start at the very top if you want, and it will show you, okay, how many of my entities under cellular processes or under metabolism regulation have these particular features. And as you dive down, you can maybe zero in on an area of particular interest to you, say carbohydrate metabolism, for example. And I can do that either via the diagram here, or I could do it via the event hierarchy on the left-hand side as well. And then if I dive into a particular piece of data, like fructan biosynthesis, there we go, it'll show you exactly where the hits are for your expression. And again, because this was a time course study over 48 hours, you'll see that there are multiple columns across the bottom, and very minor, it will be difficult to see over a web demonstration, very minor changes in coloration on this particular value. But if I do highlight the participating molecules and I work back through the time course study, you can see the expression level changing. It's just that the expression level changing in this particular random example I just picked is in a narrow band. But you can certainly look for examples of higher contrast in expression data as you work across. All right, in the second example, working from some drought stress data, this is an example of what might be a more targeted use of your experimental data. Say you're working in an area where you would expect to be the effect of your experimentation to be a little bit more narrow and only affecting certain areas of biochemical behavior. You're not sure exactly where, but once you have your data, you could bring that out here and see where that appears on the plant reactome, and that can give you a sense of exactly what areas your gene activity may or may not be occurring in, and that could lead to new targets of investigation, theoretically. So as an example of that, I pulled some data from another paper, this one having to do with response to drought stress. So comparative transcriptome sequencing was done of tolerant rice introgression lines and parents, and it took a look at how they responded to drought stress. If I grab that data, in this particular case, a set of LOC identifiers for rice and a 1-day and 3-day time course study, and I upload that data, then you see that it's perhaps has narrower hits in our curated set of data, but you could go and you could look for a particular example and zero in on that. Say, for example, strigolactone signaling down here. It's only got one hit, but that may have been a hit that you were not expecting to see, and you could go see exactly, all right, what molecule is this, and what are its values, and how are they changing between the 1-day and the 3-day measure. And then you could dig into that a little bit more closely. You could go look up D14L, for example. We'll come back actually to that example for another demonstration in a moment. Any of these results are also available via download. I just want to show you in the lower portion of the details pane here that you can download these results as CSV files for further analysis with your own independent tools or for use in manuscripts. The other main analysis tool that I want to show you today is the, let me close this box, the species comparison tool. So again, if you go back to this analysis tools icon at the top, there are two main windows. There's the overlay analysis tools, and then there's species comparison. And right now we're working off of one reference species for the plant reactome, from Rhiza sativa. And you can compare that holistically to another species of interest. For example, I will pick maize again. And what it gives you is another visual overlay that shows you exactly where there is presence or absence, as it's able to infer it, between the two species. So we're looking at the rice pathway right here for strigolactone signaling. But on that you'll see yellow and blue coloration. And what that represents is that the yellow are homologs that have been found in maize when the species comparison is done with ornithology data. The blue means that it was not present in maize. And other items that are white they're not able to infer. They're typically small molecules, or in the case of a complex like this one where you've got a partial blue and white, you look at the participating molecules. The reason is because the substance strigolactone is the other item. So that can't be inferred as present or absent between the species. It's just focused on the gene products. So again, yellow is found, blue is not found. And that can give you a sense of the relationship between the two species. We can look at another one, a larger one, for example, like steroid signaling, and you can get a good sense of where there is a comparison to be made between rice and maize. I could even turn off this overlay and show you that while I'm on steroid signaling, if I switch to the maize view of that pathway, you'll see how many aspects of the pathway disappear. If they're not able to find presence of catalytic activity in the orthology data, then those areas of the pathway are not projected. Remember, we're working from rice to other species. And so you can also move around this way amongst various species and see exactly how much of a relationship we have in our primarily comparative data between rice and some other species. And with that, I would like to end the demonstration and thank the Human Reactome Project team at OICR, NYU, and EBI, as well as the Atlas Expression team. Actually, I should mention, let me switch back to rice. There we go. I want to highlight the Atlas Baseline Expression integration. The Atlas Expression Project, they've set up baseline expression studies in rice and Arabidopsis and other species like Brachypodium for tissue type is one way of looking at it. And we've got an update to this feature coming very soon where you can actually isolate by tissue type via a diagram on the left-hand side. But you see here in the columns, you have callus, root, shoot, leaf, etc. across our various rice gene products. And then you can also look at their relative value and display the levels. There we go. Look at the numeric levels in the baseline experiments provided by Atlas. So watch this space. There will be more integration with the Atlas team. And I'd like to thank them for working with us. Lastly, thanking my colleagues at Gramene. And would just like to highlight that this is a NSF-funded project as part of the Gramene effort. And I'd be happy to take your questions via the chat window. There's been a massive rolling conversation going on over there, so I probably would have a lot to catch up on. But let me work back through and see if there are any particular questions that have not been addressed by Pankaj and Sushma as I've been presenting. Okay, so it looks like Lena's advantages, disadvantages questions have been addressed. And I don't see other questions. Laurel Cooper's wondering if there's ontology information linked to the data. There is gene ontology information linked to the data. Every catalyst in a reaction is defined by Go biological process. That is the first one that comes to mind. Any other questions? Looks like we might have a question coming in from Lena. Sure. Pankaj, you'd like me to demonstrate how we jump from rice to another species within the same pathway? Absolutely. So selecting, say, another one of our signalings that I haven't talked much about today, auxin signaling. We're looking at it here in rice. We were, just a moment. Zoom in on that. And then if I were to switch to, say, Horizon Nivara, okay, we have hits all the way across in Nivara. Let me select another one where it might just be partial. Let's look at it in Maze. Now this one looks pretty well characterized. What does change when I switch between species is the content of the gene box itself. So I'm on Triticum rartu right now, and if I select a particular gene product involved in a reaction, you see down here we have the proteins listed for Triticum rartu right here. And I could, we're still working on linking up some of this data in all interface points, but if I went back to, say, the search interface and just took a protein identifier, say I was interested in this from the get-go, then it would find a hit and it would find it in Triticum rartu, and I could go down into that search information and see that there is a reference entity for that, and I'm, with fingers crossed, if I go over to Ensemble, I would hope that that gene identifier would be found. It's attempting to load right now. Bang, there we go. So we have, you're able to link across to our ensemble data at gramene.org for that particular gene. Lena, if I start with protein identifiers that are not rice, will the data analysis tool show the result? Yes and no. It will show the results in the detail pane at the bottom of the pathway browser screen. We have a bug that we are currently addressing right now where the overlay painting on the visual diagram is not appearing for non-uniprot identifiers. And as soon as we get that addressed, then it will work across all the projected species. But for the moment, if you wanted to upload data for your species via data analysis, you would get the results in the bottom. You would also get the summaries on the left-hand side with the parenthetical entry and the statistical information. It's just lacking all of the coloration on the diagram because it's looking for a slightly too narrow set of identifiers. We're working to address that now. Dana Freund is asking, this is all projected from annotation of genes, correct? It looks like Pankaj is furiously typing an answer. We could play competing answers and see what happens. The projections are from the annotation of genes via the compare method primarily. So no, we don't necessarily provide the reference citations as Pankaj mentions, but we do have confidence levels working off of the compare data, and we also added our own further cutoffs. We put a floor of compare identity, reciprocal identity of, I think it was 40% for monocots and 30% for dicots. Actually, the other way around, yes. And which puts even more stringency on the compare data that already exists. And if there are no further questions, Dana is digging into the confidence levels. Perhaps I should just let that conversation continue for a moment. All right, we'll let Sushma get her last reply in there. And if you have any further questions, please feel free to contact us via Gramene.org. You can post questions there. That we also have an email address on the Reactome site. We've released this recently, and as with any nascent software project, there are always bugs and issues to address. So if you come across something that doesn't seem to be working quite right, please shoot us an email and we will address that. I will show you the email that you can use right here, reactome-curator@gramene.org, and feel free to contact us with any further questions. Anything else? Well, you are very welcome, and thank you for coming to this presentation.